The title of this study is Relationships of Behavioral and Physiological Symptoms of Pre-Slaughter Stress to Beef Longissimus Muscle Tenderness. The group members are Preston Ridlinger, Graham Rossi, Anna Wachter, and Alexis Zaborniak. Cattle can be negatively affected in the handling and transporting of the animal prior to slaughter. Hands-on moving of cattle can stress the cow out and cause behavioral changes in the animal. Thus, this can affect the meat quality. Animals react to the routine pre-slaughter handling differently, so this can give a wide variety and range of different behaviors that can be tested. Animals sometimes do not show a change of behavior when they're being handled, but most show behaviors such as nervousness, fear, running from the handler, balking, avoiding the handler, vocalization, aggression, or any physiological change or sign of stress in the cow. When the cow is stressed, epinephrine and norepinephrine, which are neurotransmitters, are released. When epinephrine and norepinephrine are released, they cause increased blood glucose levels, an increase in the breakdown of glycogen in skeletal muscle cells, breakdown of fat in adipose tissue, and they cause dilation of blood vessels in skeletal muscles and cardiac muscles. All of this just basically means that there will be an increase in the heart rate, and there's a breakdown in glucose and fat in the skeletal muscle cells. All of the effects added together lead to the decrease in meat tenderness. It's also documented that pre-slaughter stress affects muscle color, firmness, pH, and water retention in the muscle. The study Relationships of Behavioral and Physiological Symptoms of Pre-Slaughter Stress to Beef Longissimus Muscle Tenderness was conducted by Gruber, Tatum, Engel, Chapman, Belk, and Smith to gain further insight into the relationships of pre-slaughter stress on the quality of meat. The study was conducted on 156 calves from a single herd from the Eastern Colorado Research Center. The herd was made up of 79 steer calves and 77 heifer calves. The calves started out by being immunized and weaned. They were then sent off to Colorado State University's Beef Research Feedlot for the finishing process. The cows were separated by gender once they arrived to the feedlot and went through a period of acclimation before any tests were run. The calves were weighed individually to determine weaning weight, and then they were implanted with trenbolone acetate and estradiol. These together would increase muscle growth and appetite. The calves were then separated more, now into 16 different pens for finishing, and all 16 pens were fed the same diet, and they were fed ad libitum, which means that they had free reign of the food. Finally, the cows were almost ready for slaughter. They were implanted once more with the same substances as before, which were trenbolone acetate and estradiol. There were behavior responses collected in 3 different situations. The first one was to have a human go in the pen with the animal and take a score as to what the behavior of the animal was when that human entered the pen. The second was a reaction of the animal being confined in a squeeze chute. And the third was a reaction of being transported to the packing facilities. These animals' reactions were recorded from a scale from 1 to 5. 1 being calm, 2 restless, 3 is nervous, 4 flighty, and 5 aggressive. Blood samples were then collected before slaughter, after slaughter, and post-mortem. Also, a Warner-Brotzel shear force score was acquired. These scores together, along with other testing techniques, allowed scientists to determine the effect of handling on post-slaughter meat quality and tenderness. Now we will discuss more methods and materials in this study.
Within 1 month of the slaughter, the cattle were evaluated on a scale that Alexis talked about, 1 to 5, by 2 separate evaluators who did not discuss with each other so that the scores were unbiased. And then those 2 scores were averaged for one final score. The scores of each of the cattle in each pen were also calculated, and then one final number was found for each pen. The animals were scored within 3 hours so that there were no outside external stimuli for effect on the score. The cattle were all scheduled on 4 separate days within 90 to 111 days of the implant of estradiol that, again, Alexis discussed before. So before each designated slaughter day, 4 to 5 of the animals representing each sex and sire were tested in a hydraulic squeeze chute, and the behavior was recorded when the sides were pressed together in the chute. Blood was drawn quickly after this, and the collection in 10 milliliters non-heparinized tubes for cortisol collection. The heart rate and the temperature of each animal were also recorded for further data to see other psychological effects of the stress caused by these stimuli. The blood sample collected was about 10 milliliters, and it was allowed to coagulate at room temperature for about 4 to 6 hours and was then centrifuged. The animals themselves were then transported about 64 kilometers to a commercial facility slaughter site, and they were tested again for their reaction to human presence in the holding pens. So again, there were 2 evaluators for this portion. They went in separately and did not speak to each other, rated the animals, and then averaged a score for each animal and for each pen. The animals were then slaughtered, and blood was taken and chilled from the carcasses. The carcasses themselves then went through a series of electrical stimulation, which actually you use electrical stimulation to allow for more meat tenderness because it can rip the muscle apart, making the meat softer. So they went through a 16 volt, a 20 volt, a 24 volt, and a 28 volt electrical stimulation, and then they were sent to cool off, being sprayed for 2 minutes every 10 minutes with just some cool water. And then the marbling assessment were averaged for each carcass, with 300 being only slight marbling, 400 being small marbling, and 500 being modest marbling. Also, the tests were completed for the L, the A, and the B, which is something that's used to measure color. So that is where you take the spectrophotometer, calibrated with black and white tiles in order to compare, and then they were operated in a 2 degrees Celsius atmosphere. The strip loin was removed from each side of the carcass, or from the right side of the carcass of each carcass, and was transported to the CSU meat laboratory, where they were randomly assigned different sampling techniques to identify different longissimus muscle. After the aging process, the samples were cooked on an electric conveyor grill for a constant time of 6 minutes at 163 degrees Celsius for the top and bottom heating plates to achieve a peak internal temperature target of 71 degrees Celsius. Then the Werner-Bratzler shear force test was used on each of these pieces of meat. And this is an example of one of the machines. Basically, it's a sharp, thin piece of steel, and you put a piece of meat in the middle, and you put pressure on the thin steel, and that's measured in either kilograms or pounds, depending. And depending how much weight it takes to shear off a piece of the meat can show you how tender the meat is. So usually a lighter weight tends to mean more tender meat, and they use this to decide how palatable the meat's going to be in the end. So you usually want it to be a lighter weight. So now let's hear from Graham Rossi about analytical procedures and results.